I'm Daniel Levine, and this is the Bio Report. The next blockbuster drug may already be in production by bacteria within your body. Bacteria produce unique chemical compounds that are bioactive and may have great therapeutic potential, but this chemical space has largely not been mined. Empress Therapeutics, a Flagship Pioneering company, is searching for such molecules by exploring the genes of bacteria that live in the human body to search for the molecules they produce and identify compelling drug candidates. Since its founding in 2020, the company has already identified 15 novel candidates. We spoke to Sabrina Yang, Chief Innovation Officer for Empress and a principal at Flagship Pioneering, about how exploring the bacterial metagenome in the human body can point the way to the next blockbuster drug, the role AI plays in its platform, how this approach can accelerate the time and reduce the cost of drug development. Sabrina, thanks for joining us.
Thanks, Danny. Great to be here.
We're going to talk about the microbiome, Empress, and its efforts to look for therapeutic compounds by scouring the chemical landscape within the human body. Perhaps we can begin with the microbiome, which a number of drug developers are looking to for potential therapies. Can you start with the microbiome itself and the rationale for why this might be a rich source of new therapies?
Sure. Over the past decade, there's been tremendous efforts understanding the roles of human microbiome in health and disease, fueled by advances in metagenomic sequencing, computing power. In fact, just last year, the FDA approved the first oral microbiome therapy to prevent recurrent C. difficile infection. It's clear that these trillions of microbes that co-evolved with us play a beneficial role in biological processes, not just in maintaining a resilient ecosystem to combat infections, but also in inflammation mechanisms, modulation, energy metabolism, and so on. However, Empress is taking a different approach in that instead of starting with microbes as drugs, we focus on small molecules made by microbes inside every single one of us. Because throughout evolution, these microbes have had a chance to make, test, refine a broad range of molecules that are likely human compatible already. And over the past couple of years, Empress has built a technology platform that systematically search and prioritize the most important molecules made by our microbiome that are likely to be turned into powerful medicines.
Well, before we get into precisely what Empress is doing, I do want to call out some of the contrasts so people don't just assume Empress is the same old flavor of microbiome. Why is Empress focused only on those bacteria and their genes that occur within the human body? I mean, we see a lot of companies scouring a whole range of bacterial life for potential therapies, but Empress is focused exclusively on those within the human body.
We focus on those within the body because what are the odds that these trillions of microbes that evolved with us haven't yet made a single molecule that acts like a medicine? We already know that microbes are good at making drugs. In fact, I think more than half of FDA-approved small molecule pills were either microbially derived or natural product inspired. Of course, most of them came from non-human associated microbes. But if we're looking to make medicines for humans, why wouldn't we do so by searching for these medicinal molecules that are already in the human body? And when we started thinking about this idea at Flagship, there was little evidence of drug-like compounds found in the human microbiome. And that represents a vast, untapped, rich source of place to hunt for drugs.
So the other contrast I want to make is, we've seen companies either using things like fecal transplants or trying to devise therapies that are consisting of not just a single bacteria, but a colony of bacteria. Why is Empress focused not on the bacteria, but the chemicals they produce?
Yeah, Danny, that's a great question. So first, we believe that small molecules will continue to be one of the most valuable therapeutic modalities. They're cost effective to make. They can be taken orally. It's convenient for patients. Their versatility in biodistribution, their ability to get inside of cells to reach multiple different target classes really make them suitable for a wide range of indications. But there are many challenges in small molecule drug discovery. The time, cost it takes to find a good starting point, and then the uncertainty in safety, because most of these molecules have never been tested inside of a human until phase 1 trial, which leads me to my second point. We ask, what if nature's already done the experiment for us? What if human-compatible bioactive molecules that are already drugs themselves or can be turned quickly into drugs have already been invented by nature? And it turns out, as we were asking these questions, we started to find data that supports the idea that some microbial-made molecules, in fact, have been conserved in humans across millennia, and therefore likely have roles in health and disease. So if we put this another way, chemicals produced by bacteria inside of humans represent privileged starting points, because these are molecules that are much closer to an ideal drug candidate. And what that means is increased speed and certainty in small molecule drug discovery.
If you think of small molecule drugs, they do share certain characteristics. Listeners may be familiar with the Lipinski rule of fives. Do these chemicals produced by bacteria all fit within that world of bioactive small molecules?
What's exciting, I think, about Empress's approach is that we're identifying novel chemotypes that are not typically seen in pharmaceutical libraries, yet they're enriched for activity. In fact, I think more than half of the molecules that we tested in vivo, because we predicted them to be important in disease, we tested them in a model, they actually demonstrated in vivo efficacy. That really suggests that we're hunting in a very fertile ground. Now, back to your question about Lipinski's rule. I think it was initially set out for chemists to try and design drugs that can be taken orally by analyzing molecules that made it from phase 1 to phase 2 clinical trials. And so by definition, that means the rule is set based on what's known about existing small molecules. Mother Nature likely didn't design molecules by following those guidelines. And as it turns out, we're actually seeing both in our compound library. Molecules that fit in those guidelines, actually half of our compounds, and the rest that don't. But what we're excited about this is that so many of these molecules in our library behave like a drug without regard to whether they fit in within the rule. They're chemically tractable, synthetically tractable, they're potent, selective, stable, dose-dependent, and they appear safe in preclinical studies. Within a year since we operationalized the platform, the company's already generated 15 advanced oral drug leads.
How big a universe of potential compounds are we talking about?
We think the space is vast. We think we're just beginning to scratch the surface. But let me share 2 ways of how we think about this. First is a first principle based calculation, which suggested that there might be 10 to the 24th possible chemistries made and tested inside of 100 billion humans since the dawn of time. This is significantly larger than any in silico or physical library. In fact, it's equivalent to the number of stars in the observable universe. Another way to think about this is actually based on data generated at Empress. We've estimated that collectively inside the human bodies, the biosynthetic genes in our metagenome is equivalent to roughly 50% of the human genome in size. put it another way, about a meter long of non-human genetic DNA inside of us, waiting to be decoded and turned into potential medicines.
And to clarify, that's exactly what you're looking at. You're looking at the metagenome of bacteria to understand the chemicals they synthesize rather than just scouring something like patient blood or bacterial libraries for chemicals. What's the case for the approach, and how are you going about identifying small molecule candidates from bacterial genes?
Yeah, it's a good question. Most chemicals in our bodies have not yet been characterized. So the challenge really is, how do one focus on the right ones to characterize? Because it's simply practically impossible to take a brute force approach given how vast the space is. And so 7 years ago, when we at Flagship started to think about the idea that became Empress, one of the first questions we asked ourselves was, what if bacterial genes in the human body hold the answer to the most drug-like molecules? Why? Because differential metabolomics, chemical first approaches, they tend to focus on the most abundant and therefore high concentration, i.e. high concentration molecules. So by definition, they're the least likely to be potent. A genetics first approach allows us to find molecules that are more likely to be drug-like. It allows us to find molecules that may be in lower concentration ranges in our bodies, but by definition, higher potency. And they're mostly ignored by mass spec or chemical first approaches. And to your question about how, I think there are multiple different innovations that have to come together to enable what we do here, starting with the metagenome to find drug-like molecules. But I will say that one key component is our ability to use AI to quickly scan through large amounts of bacterial DNA to predict the ones that encode for biosynthetic enzymes. And then these enzymes actually work together to synthesize or modify bioactive molecules. So we can now find small molecules with a genetic basis in the body. And then these genetic associations then point us to disease relevance.
You've developed what you call your Chemologics platform. What's the starting point from this? Are you using public databases of bacterial genetics, or are you sequencing bacterial genomes?
Sure. First of all, maybe I can explain why we call it Chemologics. It's actually quite literal. The platform combines the versatility of chemistry with a genetics-based programmability and inherent human relevance of biologics. So Chemologics. What really revolutionized our industry and our foundational to what we call biotech today is the sequencing of the human genome and recombinant DNA technologies. It's created billions in value, it's impacted millions of lives. And what we at Empress realized is that the very same concept can now be applied to small molecules. Our platform focuses on bacterial DNA in the body from both public and private data sources. And we use AI to predict genes that are most likely to encode compounds that play a causal role in health and disease. Then we recode these genes using synthetic biology, engineer microbes to actually produce these compounds for us so we can characterize their structures. But because we infer the chemical identity from DNA in a computer before any wet lab experiments are even done, we can actually focus our search on the most drug-like compounds that are synthetically and chemically tractable, molecules that are amenable for metchem cycles and cost-effective to scale up. And finally, perhaps most importantly, our process does not start with a target. Instead, it starts with a bioactive molecule with a genetic basis. We use a suite of innovations in AI, functional genomics, chemoproteomics to reveal potential targets and mechanisms through which these molecules exert pharmacology. So what this means is the potential to develop first-in-class programs for many indications where new targets or mechanisms are needed to address unmet need.
At what point do you actually synthesize a molecule, and a typical drug discovery operation, I imagine, would build a library of these and just screen them against targets of interest. It sounds like you're not doing that.
Exactly. Yeah. Like I said earlier, one benefit of starting with patient metagenomics data is that at a very early stage in our process, we can prioritize the most promising molecules to pursue. So for each indication, just an example, we prioritize tens to hundreds of molecules to synthesize. And in this process, we've already identified 15 drug leads. For comparison, if you think about traditional small molecule drug discovery that starts with a target and a hit screening campaign, that often requires orders of magnitude more molecules synthesized before even one lead candidate can be identified. And the beauty of a genetic chemistry forward approach is that it allows us to find bioactive molecules first and then allow it to reveal targets and mechanisms that they have evolved to modulate our biology. And so every time we have a target in hand, we already know it's druggable, and we already have a hypothesis about why drugging it would matter in a disease. And when it comes to thinking about therapeutic value, I think if you think about human compatible molecules that exert its effect through novel disease relevant targets, it really is first in class opportunities. And in some cases, our molecules lead us to targets that appear to be relevant in multiple high impact therapeutic areas. And that creates a pipeline in a pill.
You say you've identified 15 lead candidates. Is there any relationship between the class of therapies they represent, their targets or indications?
Yeah, I can share that because these 15 molecules came straight out of our platform with actually very little to no medicinal chemistry required. And many of them have already been optimized by bacterial medicinal chemists inside of us. They look like a drug, they act like a drug. There's a broad range of target classes that they engage, from GPCRs to enzymes inside and outside of a cell to carrier proteins. And I can share that we're initially pursuing immunology and inflammation, where there's massive unmet need to find safer oral medicines that work better for patients. I think this platform can be broadly applicable to so many indications and disease areas beyond I&I, including oncology, metabolic, and neurological disorders.
And what's the plan for moving any of these to the clinic?
Well, we anticipate to be in the clinic next year. And we're also in conversations with global pharmaceutical companies about potential asset-based or discovery collaborations, really to leverage our platform and bring more innovative therapies to patients faster.
The case for your approach is not just the potential to explore and uncover novel compounds that have therapeutic benefits, but to do so faster and more cost effectively than conventional drug discovery approaches. Can anything be said yet about the advantages your approach provides? Is there any way to quantify the time or cost?
Instead of 4 to 7 years from start of a program to a development candidate, Empress is actually on track to cut that discovery and preclinical R&D timeline down by 50%. From a cost standpoint, on average in our industry, unfortunately, it still takes $1 to $2 billion to develop a drug. 90% of drug candidates fail in the clinic. Empress's approach, by starting with molecules in the body, co-evolve for health and disease, that we think what that means is the potential to achieve higher clinical success rates due to the inherent human compatibility of these molecules and the co-evolved targets that are likely pharmacologically relevant. Of course, this will have to be proven out in the clinic. However, if our assumptions are correct, then I think this approach will have significant effect on cost, time, certainty in small molecule drug discovery.
Empress is a Flagship Pioneering company. Flagship provided an initial commitment of $50 million. I suspect there are a number of other platform technologies in the Flagship portfolio that you might be able to leverage. Is there anything Empress is doing to leverage other technologies in the Flagship portfolio?
Absolutely. Flagship has a long history of originating and investing in microbiome-based companies. In fact, the world's first microbiome-based oral pill approved by the FDA came from Empress Therapeutics, which is founded by Flagship in 2011. And since then, Flagship's created multiple companies in the space, ranging from microbial cocktails to monoclonal microbes to prebiotics to modulate microbial metabolism for human health, and now to Empress, where we're taking a small molecule approach to find mechanistic clues as to how the microbiome exerts functional effects on human biology. So we've benefited tremendously from the wealth of knowledge that Flagship has accumulated over the past decade. And we're continuing to engage with our ecosystem companies as we explore new technologies and expand our platform.
Sabrina Yang, Chief Innovation Officer at Empress and Principal at Flagship Pioneering. Sabrina, thanks so much for your time today.
Thanks, Danny. It's a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.